Now we will see the microbiology of yogurt, and we will discuss the microbiology that is involved in the production of yogurt. If you look at the process and the steps involved in the process of fermentation or making the end product, it is an 8-step process in which we first process the milk, we add starter culture, we look at the growth of these microorganisms. The microorganisms produce metabolites, they produce flavor compounds, they make formate product, they make slime in the end product, and then proteolysis. So these are the reactions that they take place while the microorganisms grow on during the process of making yogurt. So milk treatment, as we said, that if we have to have a controlled fermentation process, we need to control the initial load of microorganisms. So that is why the milk is first heat treated or pasteurized, or sometime it can be sterilized, it is homogenized and standardized, so that we know the exact composition of milk. It is controlled, it is heat treated or sterilized, so that the other microorganisms do not show their activity during the fermentation process. So milk has to be treated. Then we add starter culture, and we earlier discussed that it can have Streptococcus thermophilus or Lactobacillus species that can be added in combination. The next, again, there may be other types of microorganisms or bacteria can be added, like acidophilus or Bifidobacterium, Lactobacillus casei, rhamnosus, etc. So these can be all added up. It depends upon the product, but these are generally reported to be added in many of the different kind of products. We have to look at the combination of Streptococcus and Lactobacillus ratio. Ideally it should be 1:1 ratio, and it should not exceed 3:2. So because they perform different functions, and every strain or every bacterial strain would have different functions to be performed within the end product. So we have to look at the level of activity and the end product metabolite that have to be produced in the final product. So that's why we need to control their ratio as well if we want to have a consistent end product. Now the next step is we have to look at the conditions of growth of microorganism. Especially in case of yogurt, we would prefer that this fermentation process takes place at an optimum temperature, that is 43.3 degrees C, in which the acid producing bacteria as well as flavor producing bacteria would have an optimum activity, because we would want the end product to have a certain amount of acid as well as a certain amount of flavor that has to be produced within the end product. So just in case if the temperature goes above this 43.3 degrees C or 110 degree Fahrenheit, the Lactobacillus species would dominate, and this Lactobacillus species produces acid. So that would mean that we have made a product that have got higher acid content. So the product is more acidic, it is bitter in taste, and it does not have the flavor component in it. So the final product would not be desirable. That's why we would prefer that this temperature of fermentation must be controlled. Lactic acid is produced, as we said, that from the lactose. Once it is broken down, the lactic acid is produced, and then there are flavor compounds within yogurt, such as aldehyde. It has to be 25 ppm, and some of the diacetyl up to 0.5 ppm, they are desired in the final product, so that we can have a desirable flavor profile of the yogurt that is produced. Formate production. Formate, again, it is necessary for the growth of lactobacillus, and it is produced by Streptococcus thermophilus from pyruvate in action of formate lyase, that is an enzyme that would be making up formate, and that formate has to be produced in order to lactobacillus would only grow if this is enough formate present within the substrate medium. So this is how one microorganism is dependent upon another microorganism for production of certain products that they consume for their normal metabolic functions. Then we also look at another reaction called as slime formation or glycan formation. As we earlier said, that these glycans are the carbohydrates. So beta-galactosidase from some strain of Streptococcus thermophilus, they would be polymerizing these glucose to make up a little complex structure called as oligosaccharide or glycans. Particularly glycans would give a slimy appearance or slimy feeling to the product, so the texture would be desirable. The product becomes viscous in nature. So this is what is desirable in yogurt, because we want to have a viscous product at the end. And the last reaction is proteolysis. In case of proteolysis, the proteinases from lactic acid bacteria, they would be breaking down the milk protein, and these milk protein would then again add up to the flavor profile as well as the taste profile of these milk which are used for fermentation. If you look at the problems that can arise in case of yogurt, so as I earlier said that the temperature would have an effect of growth of one organism or the other, and they would also affect the end products. So there are chances that there might be higher concentration of this acetaldehyde. If we have a lower concentration of acetaldehyde, we can have a chalky flavor or a sour flavor that comes in. And if we have too much acetaldehyde produced within the end product, we can get a green flavor that comes from the end product. So this would not be desirable. Similarly, if we have too much diacetyl produced within the end product, so we would have a buttery aroma that can be desirable. And if we have too much acid production within the end product, so the product would be sour in nature. It would not be good tasting because of this high amount of acid present. Similarly, proteolysis reaction, as we earlier discussed, it breaks down the protein compounds into a simpler components such as peptides, and these peptides can also change the flavor, and they can also make a bitter product, which is highly undesirable end product. So this was the process for manufacturing of yogurt and the microbiology involved in the manufacturing of yogurt, and this tells us that how important it is to control the factors such as growth factor as well as the ratio of these microorganisms to have an optimum condition in which they can collectively work on this substrate, that is milk, to make up an end product, yogurt, which has got the desirable attributes.